Darwin's finches and the ALX1 gene. More than a century ago, Charles Darwin visited the Galapagos Islands and noticed morphological differences in the beaks of various finches across different islands. This began his theory of evolution by natural selection. The classical taxonomy he came up with used purely morphological features to determine phylogeny. That is, Darwin simply used physical observations to determine the different species of finches and trace their ancestry. Adaptive radiation is when organisms diversify rapidly into new forms caused by changes in the environment. Morphology is related to physical features present in an organism. Phylogeny is the history of a species' evolution. In this picture, you can see the roughly 15 species of Darwin's finches that have propagated from a single ancestor and their geographical origins. Different colors indicate different species of finches. In a recent article published in Nature, the genomes of 120 individual Darwin's finches were sequenced. The branching order of recently diverged species was determined, and discrepancies between the historical and modern phylogenetic trees were found. The findings proposed a revised taxonomy for the sharp-beaked groundfinch and the large cactus finch, the radiations of which began roughly 100 to 300,000 years ago. A haplotype was found to be strongly associated with beak shape, including one specific gene, the ALX1 gene. A haplotype is a set of DNA variations that tend to be inherited together. The ALX1 gene encodes for craniofacial development, most notably beak diversity. This adaptive radiation occurred at great speeds due to isolation, formation of new islands caused by volcanic activity, changing sea levels, and the El Niño phenomenon. This diversity from gene flow allows finches to expand their utilization of food sources.
To create a phylogenetic tree based on mitochondrial DNA sequencing, blood samples were collected from 200 of Darwin's finches, including the 14 species that reside in the Galapagos and one on Cocos Island. The finch's DNA was then isolated using a DNeasy tissue kit. Then, the genome sequences of these 200 samples were read against that of a female medium ground finch as a control for comparison in order to set up the phylogenetic tree. For dating the phylogenetic tree, they used a previously found mitochondrial DNA mutation rate of 2.1% per million years and found that the divergence of the finch species began around 1.5 million years ago. Research allowed for a newly dated phylogenetic tree to be created, which indicates the presence of adaptive radiation in the finch species within the past million years. The evolution and diversification of the finch species was caused by extensive gene flow and hybridization, which influences the evolution of the different beak shapes. A comparison of the genetic differences of the finch species highlighted the ALX1 gene as being significant in the beak formation of Darwin's finches.